Hello, everyone, and welcome to today's webinar, Cell-Based SPR Microscopy Application in Drug Discovery. I am Cassie Saltman of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Biosensing Instruments. To learn more, visit www.biosensingusa.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Shijie Wu, Application Scientist, Biosensing Instrument, and Dr. Marie-Laure Rives, Head of Biology, PostEra. Dr. Wu, you may now begin your portion of the presentation.
Thanks for the introduction. My name is Shijie Wu from Biosensing Instrument. Today I'm going to talk about the basic principles and applications of surface plasma resonance microscopy. Before we start talking about SPRM, please allow me to say a couple words about Biosensing Instrument. Biosensing Instrument is located in Arizona, USA, and we started in 2005. So since 2005, we have developed a series of platforms for specialty SPR measurement. Particularly in 2017, we developed this new platform called SPRM, which is very well recognized by the industry and received quite a few awards from different institutions. And it's also very well recognized, received a lot of interest from researchers all over the world. SPRM is a label-free technique that allows you to directly culture a cell onto the SPR chip surface. Therefore, you can study the binding kinetics with membrane proteins directly on the cell surface, close to their physiology conditions. It can provide a real-time analysis and give you the binding parameters, including the association and the dissociation rate constant and the binding affinity. It can also be used to study some nanostructures and nanoparticles, including species like virus, bacteria, and so on. SPRM has three key components. The first component is the SPR unit. It includes the light source, the detector, and the sensor chip. On top of this SPR unit, it is a bright-field optical microscope, which essentially illuminates the same area, or looking at the same area. This optical microscope allows you to precisely position and locate the sample to where you want to do the experiment. It also allows you to monitor the status of the sample during the experiment. The last component is a uniquely designed flow system. And this uniquely designed flow system provides the optimized flow conditions to ensure accurate SPR measurement. It also allows you to directly culture the cells onto the chip surface and maintain the measurement conditions during the experiment. By combining all three of the components, and the system allows us to measure the parameters of the binding kinetics, including Ki-on and Ki-off, and the binding affinity. This is how optical system and the chip well looks like in a real system. The optical microscope looks from the top of the chip. The polymer well sits on top of the gold chip, and the gold chip can be functionalized chemically for culturing of different cell lines. Most commonly, they will be used like collagen, PLL, and PDL, and etc. After the experiment done, the data will be saved in a series of images, including the bright field and the SPRs. And then we'll divide the whole area into a number of small regions we call the region of interest. So usually it will be around like 600 up to several thousand, depending on the time you want to spend for the calculation or the computation. And then for each of the ROI, we'll generate a sensorgram. So most of the time, the experiment is done in kinetic titration kind of fashion. So which means it will go from low concentration to high concentration, just continuously injecting one after another. And once we have all the sensorgrams generated, and then we'll feed those data to the one-to-one binding model. And then we'll apply the binding filter and to remove all those non-specific bindings and leave only the good specific bindings corresponding to the molecule and the receptors on the cell. And from those, we can get a histogram distribution, and which will give us the average K-on and K-off, and also the affinity. Now I will show you a number of examples for the binding measurement using SPRM. So this is one of the nice experiments we've done with AstraZeneca. And the system we are looking at is a small molecule, AZ1395, targeting the GPR39 receptor on HEK cell. The result is shown here on the left. This is a SPR image, and on the right is a corresponding optical image. And the optical image, you can see more clearly where all the HEK cells are located on the chip surface. And each of the color square on the surface reflects one of the good areas, give you a good binding data. And you can see immediately the good binding data overlaps with the position of the cell very well. So this tells us we are looking at the bindings happening on the cell, not on some empty areas. And with all the data we analyzed, we can obtain the isotherm from which we can obtain the binding affinity based on the thermodynamic analysis. We also have all the sensorgrams for each of the unique area, and we can feed the kinetic model, the one-to-one binding kinetic model for each of the sensorgram, and from the fitting we can obtain the value, the average value of the K-on and K-off, and also the binding affinity. And also we see the, because we are looking at many different areas, and we get a distribution histogram which shows the variation or the heterogeneity between the different cells or the different parts of the surface. So the results we obtained for the binding affinity is about 400 nanomolar, and the corresponding K-on and K-off reported also here. And this molecule is actually very well studied by AstraZeneca and other researchers. So one of the results reported in the literature using a technique called the BSI, which is the backscattering interferometer, and it gives a value about 233 nanomolar, which agrees very well with our measurement down here. This is another small molecule example binding to the membrane transporter, and it's the data from Casey. And in this experiment, we have done positive binding and also negative control samples. And you can see for the positive experiment, we get this nice binding data. And for the negative, essentially no bindings observed on the cell. This slide here shows a comparison between the affinity measured on the purified protein versus cell-based protein. And this cell-based protein measurement is done by signature using SPRM. Again, the result is shown on the top part of this slide. And again, in the middle, that's the optical image of the cells being measured. And it shows the location, the position of the cells on the slide, and also those squares, which is the ROIs,
indicates where the binding occurs. It actually overlaps very well with the position of the cell. On the right, that's the SPR image, which is measured during the experiment. The left side is actually a fluorescence image that verifies the overexpression of the ATS1 receptor on the cell. The measured result, or the affinity between this compound and the ATS1 receptor using SPRM is about 2 nanomolars. And this ATS1 receptor being purified. And it was actually measured using a conventional SPR. And this work is done by Dr. Huber from Roche in Basel, Switzerland. And Dr. Huber actually purified the protein and immobilized them on a sensor and using traditional SPR. And she measured the affinity between this compound and the ATS1 receptor is about 400 picomolar. And there's also some works involved using in vitro and in vivo data using cell-based work and using human type and wild type cells. And the measured result for the wild type is about like 8 nanomolar, and well, the other one gives about like 500 picomolar. So apparently we see some difference between the purified protein and the cell-based work. Actually, we have seen there's a general trend. The affinity measured with the purified protein is usually shows a stronger affinity compared to the cell-based measurement. So overall the results agrees fairly well between the different measurements using different techniques.
This is actually a very interesting example. In this case, we have a bivalent antibody and binds to the BR's results. And this is data is from Genentech, and we actually have a very good webinar with Elaine Mai, and she gave a very good presentation. If you are interested, you can go to the webinar. And what is interesting here is for the bivalent antibody, and we do see 2 binding peaks.
So far, the examples I have been showing you are mostly with the adhesion cell, and also they are fixed. So many people have been asking for whether we can work with suspension cells instead of adherent cell, and also can we work with live cells instead of fixed cell. So first of all, for the suspension cell, in the beginning we do have some difficulties because we don't really know how to fix them onto the surface or keep them on the surface, because they tend to flow in the buffer solution. Fortunately, with the protocol developed by John Brooks from Pfizer, we'll be able to grab them from the solution and keep them on the surface and to study them. So this example here shows the binding between rituximab and the live suspension B cell. Rituximab is the first antibody approved by FDA for treating cancer cells. The experiment we did here using SPRM is we catch the suspension B cell, a live cell, onto the chip surface, and then introduce rituximab, the antibody, and to study the binding. So the results, the affinity we measured using SPRM is about 1 nanomolar. The affinity cited by FDA based on different techniques is from 5 nanomolar up to 10 nanomolar. So in general, our measurement on live cell with SPRM agrees very well with the other cell-based techniques.
This is one of the latest experimental results involve SPRM, which is done by John Brooks from Pfizer. And this experiment studies the direct binding between a human camerin and to the CCRL2 receptors on a HEK cell. The CCRL2 is a non-signaling receptor that binds chemotactic ligands to shape leukocyte recruitment to sites of inflammation. However, there have been a lack of knowledge about what type of ligands directly binds to CCRL2, and also the corresponding functional impacts. And this study is the first time actually to measure direct binding between a camerin and the CCRL2 receptor. The experiment shows a specific binding to the CCRL2 receptor, and well, shows there's very little binding on the parental cells, which is a negative cell. The measured affinity between the camerin and the receptor is about 7.5 nanomolar. And John actually gave a very nice webinar presentation which is available on our website. So if you are interested in looking at more details about this work, you can go to our website and to download and listen to the webinar by John.
SPRM can be combined with other techniques like electrochemistry. So we can actually add a potential stat to the system, and then we can supply electric modulation to the system, and then using the SPR imaging capability to monitor the local responses of the chip surface, the materials on the chip surface, to the electric modulation. Here are two examples for the electrochemical applications with SPRM. The first one is the SPR-based impedance experiment. So what you're looking at is the responses of SEHA cell to the apoptosis treatment. And you have the optical image, the SPR image, and the SPR-based impedance image. So the SPR-based impedance image shows the localized responses of those cells to the treatment. And the second example, it shows the CV, using the CV to study the individual gold nanowires on the chip surface. So each of the gold nanowire was shown up with the interference pattern on the image, and they can be labeled from here. So for each of the gold nanowires, you can have a CV based on the SPR signal. So you can see depending on the size and the conductivities and those different properties of the individual gold wires, and they should see differences in the CV. So which proves those nanowires are not uniform. They have different physical shapes and scales and properties.
Thanks for listening to this presentation. So in summary, SPRM is a very unique technique. It's a label-free technique that allows you to measure the binding between a compound and membrane proteins directly on the cell surface. So there's no need to purify the protein as you have to do in a conventional SPR. So with this, and I will pass the time to Marie-Laure. Thank you.
Hi, good morning. So my name is Marie-Laure Rives, and today I'm going to present some work that was performed at Ferring Pharmaceutical. I'm going to talk about the pH dependence of the GPR4 antagonist that we believe impairs his therapeutic potential, and also highlight how SPR microscopy has helped us characterize its mechanism of action. So firstly, we were interested in GPR4 and more specifically in the potential of a GPR4 antagonist for IBD or inflammatory bowel disease. IBD is characterized by a chronic inflammation of the gastrointestinal tract, and acidosis has been shown to be associated with a variety of pathological conditions, including the inflammation observed in IBD. In both active ulcerative colitis and Crohn's disease, intraliminal colonic pH is significantly lower than in healthy control. But we'll go back to that later. There are several acid-sensing receptors. They are proton-activated G-protein-coupled receptors, or GPCR, such as GPR4, GPR65, and GPR68. And those receptors are pH sensors that can trigger intracellular signaling in response to alteration in extracellular pH around physiological values. So these receptors have been linked to several well-studied pH-dependent physiological processes, including IBD. But more interestingly, GPR4 is predominantly expressed in endothelial cells, and in several studies, GPR4 activation has been shown to increase the expression of inflammatory and stress responses. So you can see on the left one of the transcriptomic studies that was done in UVEC cells that shows that activation of GPR4 by acidosids led to the upregulation of a number of cytokine, chemokine, but also adhesion molecule. In another study, GPR4 activation in UVEC cells by acidosis again led to an increased adhesion of monocytic cells onto those UVEC cells. So the overall hypothesis and proposed mechanism of action is that in acidic inflamed tissue, proton would lead to the activation of GPCR4, leading to an upregulation of chemokine, cytokine, and adhesion molecule, promoting the attachment, adhesion, and infiltration of immune cells into the diseased tissue. Additionally, there were several papers showing that inhibition of absence of GPR4 could ameliorate colitis in IBD animal models. It was shown first in the DSS model, in which GPR4 knockout mice seems to display an improved body weight loss and lower histopathology score compared to the wild-type animals. But more specifically, in IL-10 knockout mice, so those mice developed spontaneous colitis, which is highlighted here by a high histological score in the IL-10 knockout mice, and also highlighted by the pictures on the right. Compared to those IL-10 knockout mice, the double knockout mice, GPR4 IL-10 knockout, seems to be protected from the development of severe colitis, which you can see here in the much lower histological score in those double knockout mice. So it was proposed that GPR4 inhibition would be a valid therapeutic approach for the treatment of IBD. Before investing in too much medicinal chemistry, we wanted to perform some proof of concept studies and target validation experiments using compound 13 that has been reported in the literature as a potent and selective GPR4 antagonist, and use this compound for a proof of concept study to verify the anti-inflammatory effect of GPR4 inhibition in vivo. And in light of the study presented here, we chose IL-10 knockout mice. Before that, we wanted to profile compound 13 in vitro and confirm its potency, but I will get back to that later in this presentation in more details. But in the mouse GPR4 receptors, in traditional antagonist mode, the potency of compound 13 was measured to be around 200 nanomolar, which is represented by the dotted orange line here, and that was pretty much in line with data reported in the literature. We also confirmed the pharmacokinetics properties of this compound in male BAPC mice, and the data were also pretty much in line with the literature. So after administration of 30 mg per kg in those mice, the Cmax was around 54 micromolar with a relatively short half-life, but the simulation led us to estimate that after BID dosing, we would still get sufficient exposure for a significant target engagement of the receptor. So we proceeded to perform a study in IL-10 knockout mice using compound 13 at 10 and 30 mg per kg BID. Unfortunately, compound 13 failed to improve colitis in this model. So first, we'll look at the number of endpoints. Firstly, you can see that the healthy control in blue gained weight during the course of the study. In contrast to those healthy control, the IL-10 knockout mice in red, they failed to gain weight, which is representative of the disease. And we chose also a positive control IL-10, anti-IL-12, 23 antibodies that has been studied extensively in those models, but is also clinically validated. And you can see that with this model, the animals gain weight comparable to the healthy control. In contrast to that, compound 13 trained mice failed to gain weight. We also measured colon weight to length ratio as a gross readout of inflammation. It's increased about twofold in the IL-10 knockout mice, but this increase is totally reversed with our positive control. In contrast to that, again, there was no significant effect with compound 13. We also looked at other markers of inflammation, such as fecal lipocalin, and we observed similar results with a lack of significant effect with compound 13. So additionally, histopathological analysis of the distal colon showed similar results. In this model, the histopathologic sum score was around 4, so suggesting that the colitis induced was relatively mild, but in spite of that, we can see that our control completely reversed the disease and the phenotypes of the disease in this study. With compound 13, in contrast to the positive control, although there was a slight trend toward improvement, those data were not statistically significant. The other two graphs represent some of the scores and measurements that were included in the sum of the histopathological score, such as inflammation score and hyperphagia score, in which we observe a similar trend with compound 13, but that were not statistically significant, although we see a robust effect with a positive control. So although the exposure data that I presented earlier suggested target engagement, to confirm those results and to confirm we had proper engagement of GPR4, and also in the hope to identify a PD marker and to confirm the mechanism of action of GPR4 inhibition, we also performed transcriptomic analysis of the terminal colon sample. So first, on the left, we just compare the transcriptomic profile in the wild type versus IL-10 knockout mice, in which you can see significant alteration in gene expression that are related to defense and immune response. In the control treated animals on the right, you can see that treatment with anti-IL-12, 23 also leads to a significant change in gene expression and a reversal of the effect seen in the IL-10 knockout mice. However, in the middle, in the compound 13 treated animals, you can see we were not able to measure any significant changes in gene expression, suggesting a complete lack of target engagement. So this Those results were very puzzling and suggested to us that in spite of exposures that were predicted to yield target engagement, we did not measure any significant changes in gene expression, suggesting again lack of target engagement, and that maybe the mechanism of action of compound 13 was a little more complex than I was originally suggested. So to confirm these results, we decided to perform a more thorough analysis of the pharmacological profile of compound 13 in vitro. And so GPR4 has been shown to couple to multiple pathways, including GS, leading to increased cyclic AMP production. So we first developed a cyclic AMP assay and profiled the antagonist activity of compound 13 at both human and mouse GPR4 at several pH. So you can see, for instance, that around pH 6.8 or 7, compound 13 is relatively potent with a potency at pH 7 of 7 nanomolar, about 100 nanomolar at 6.8, but below pH 6.4, compound 13 is either micromolar or completely inactive. We observe similar results at the mouse receptors. In addition to cyclic AMP, we also profiled the pharmacology of compound 13 at other pathways, including GQ and G13, and observed similar results with the potency of compound 13 being dramatically pH dependent. To make sure that those results were not an artifact of recombinant system, we also confirmed the pH dependence of compound 13 in UVEC cells that endogenously express GPR4. So as previously shown in the literature, in endothelial cells, GPR4 activation by acidosis stimulates the expression of a number of inflammatory genes, including here, which we were able to confirm, E-selectin and CSF2 expression is induced by acidosis at both 6.8 and 6.4 in blue and in green. However, 1 micromolar of compound 13 only led to a significant inhibition of the induction of those genes at pH 6.8, but not at pH 6.4, confirming again the pH dependence that we observe in recombinant system. So overall, those data suggested to us that compound 13 mostly behave as an orthosteric antagonist. What does that mean is that the potency was highly dependent of the agonist concentration, and basically the pH sensitivity of GPR4 was dramatically right-shifted in presence of increasing compound 13 concentrations. So that has a major implication on our exposure prediction. For instance, we assume the relevant potency of compound 13 was around 200 nanomolar in mice. But now if we take into consideration its pH dependence, not really knowing, and we'll get back to that at the end of this presentation, what is the relevant pH, that could potentially explain the lack of target engagement that we observed. So before confirming the mode of binding and inhibition of compound 13, we wanted to confirm that it wasn't specific to compound 13 and that it wasn't due to the compound being protonated in low pH condition. So we confirm the same that other analogs of compound 13, whether or not they would also be, the potency would also be pH dependent. The reason behind this was mostly because compound 13 has, you can see here, had a basic amine that could potentially be protonated in low pH condition, which could affect its affinity and potency on the receptors. So we profiled those 3 analogs at both human and mouse GPR4 in the same cyclic AMP assay that I showed you before. And you can see that we observe the same thing. For instance, for compound 39C in bright green here, it's about 100 nanomolar at pH 6.8, but as you can see in light green now, the curve is dramatically right-shifted, and it's now either almost inactive micromolar. And the most interesting compound out of those 4 was compound 2, because it did lack the basic amine present in compound 13, sort of confirming that the pH dependence was probably not due to this chemotype, but most likely to the mode of action of the compounds to the receptors. And as I mentioned before, compound 13 did behave as an orthosteric antagonist, right shifting the pH curve to the right, its potency being dramatically decreased in presence of low pH condition. So we hypothesized that compound 13 could bind to the conserved orthosteric binding site. And what also led us to believe that is that compound 13 originated from a lead that was found to be also active at the histamine H3 receptors by displacing the binding of radiolabeled histamine on it, suggesting again a competitive mechanism of action. So to confirm this, we utilized a model structure of GPR4 that was released from AlphaFold. And we also utilized available crystal structures of several histamine receptors and aligned them to the AlphaFold model of GPR4 to identify residues in the conserved orthosteric pocket of GPR4 that would likely be involved in the binding of the compound 13. And we performed some mutagenesis of some of those residues in that binding pocket to see if those mutations would affect the binding and the potency of compound 13. Some of those mutations led to non-functional receptors, so that we could not profile compound 13 for those, but several of them were active with a similar pH 50 as wild type, so including tryptophan 73 and tyrosine 269. So we were able to profile the potency of compound 13 at those receptors. And that you can see that the potency of compound 13 is slightly right shifted in one of them, but especially for tryptophan 73A, the potency of compound 13 is dramatically affected, and suggesting that compound 13 indeed binds to this pocket, and that mutation in that pocket dramatically affects the ability of compound 13 to bind effectively. You will note that the presence of an histidine in this binding pocket, histidine 269, that has been shown in the literature to be one of the histidine in GPR4 proteinated and leading to GPR4 activation. So that also led us to hypothesize that the reason why compound 13 potency is dramatically affected in low pH condition could potentially be due to this histidine being protonated in low pH condition. So to confirm the binding properties of compound 13, that's when we use SPRM. So we use GPR4 expressing cells that were fixed following stimulation with different pH in the hope to fix GPR4 in either its inactive or active conformation, and then measure the ability of compound 13 to bind in those different conditions using SPRM thermodynamic analysis. And you can see that at pH 7.9 and 6.8, we do see significant binding events with estimated KD between 3.9 and 5.5 nanomolar, which is in line with our functional data. But interestingly, at pH 6.4, we failed to measure significant binding events. No binding events basically were, or very few were detected at pH 6.4, which seems to confirm my hypothesis that in low pH condition, most likely due to the protonation of a number of histidine, including histidine 269 in the orthosteric binding pocket, that in those conditions compound 13 cannot bind, and explaining the pH dependence of compound 13 in our functional assay, and potentially explaining the lack of efficacy that we observe in our in vivo studies. It's a little difficult to accurately measure the relevant pH in IBD, in human IBD, but also in the animal models. So I mentioned before that luminal pH in IBD is quite low compared to healthy control and measured to be between 5.5 and 7. But the luminal pH is likely not the relevant pH. Since GPR4 is expressed in endothelial cells, the relevant compartment is likely the mucosa, in which it's very difficult to measure exact pH. There are pH probes that have been used in both cancer and inflammation that suggest that the pH can get as low as 5.8 or 6. But due to the patchinase nature of acidosis, it's very difficult to accurately measure pH in disease, but we estimate it will likely be between pH 6 and 7. And we know that in that range, the potency of compound 13 is dramatically decreased. So we believe that in light of all those recent data, compound 13 is probably not the best tool to validate the role of GPR4 in inflammatory conditions. So I would like to thank all the contributors of these studies, and thank you very much for your attention.
Okay. Thank you, Shijie and Marie, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of the screen, and we'll answer as many of your questions as we have time for. Let's go ahead and get started here. Our first question is for Shijie. The surface of the cell has many receptors. How does SPRM detect the binding of my specific target receptor?
Yeah, that's a very good question. Basically, for SPRM, and there's two approaches to this. The first thing is you can overexpress the target on the cell surface, and so to make the target proteins available in a large quantity, and comparing to the other proteins which you do want. And the other approach is, you can always use a negative control, either use a negative control compound or use a negative control cells. So by doing this, and you can verify you are actually working with the binding between this your compound with this specific targets on the surface.
Wonderful, thank you. Our next question here, it looks like, is for Dr. Rives. Compound 13 and analogs have shown efficacy in numerous animal models of colitis, as well as other indications. How do you reconcile those findings with the current study?
Yes, thanks. It's also a very good question. So as I mentioned at the end of the presentation, it can be very technically challenging first to measure the exact pH in the target tissue. So the exact degree of acidosis in all those different models is not particularly well understood. And also the degree of acidosis is directly correlated with the degree of inflammation. So not all models induce a similar degree of inflammation. And so, for instance, the IL-10 knockout mice, they are developed spontaneously colitis, so you can also get very heterogeneous results in terms of inflammation or in acidosis, consequently, depending on how the models is designed. So I think it would likely explain the discrepancy between the different models.
Fantastic, thank you so much. Our next question here is for Shijie. What is the range of affinities that can be measured by SPRM?
Yeah, the SPRM is a SPR technique. It's actually behave, the similar to like a conventional SPR, except for the the measurement is doing differently using an imaging technique, and the sensitivity for the SPRM is actually comparable to conventional SPR as well. So it ranges from picomolars up to micromolars for the affinity.
Thank you. Our next question here looks like is also for you, Shijie. How many nanometers for cell's thickness can be a sensing area?
Oh, that's actually a very good question. Yeah, so I think the people who's asking this question probably knows conventional SPR pretty well. For the conventional SPR, one of the concepts, is the measuring depths or the penetration depths of the emission light waves. However, it usually says, the SPR can measure up to a distance about 200 to 300 nanometers above the chip surface. However, with the cell, we are actually, the measuring mechanism is quite different. The cell is actually a dynamic system, so it's kind of like a life system. So what happens here is when you have a compound binds to the cell surface, and then the binding will cause some changes in the cell, including the surface tension and morphology and stiffness and those kind of physical chemical properties. And all those changes in the on the cell will be translated to SPR signal, because those changes will cause the dielectric properties change around the cell's area. So the SPR essentially is actually measuring the dielectric property changes on the interface. So that's why even the cell is actually could be very large, tens of micron, maybe 100 microns big, but the binding on the cell surface can still cause a change in the dielectric property, and we can still measure the changes caused by the binding. And that's how we actually use the SPR to measure the kinetic binding between compound and membrane proteins.
Thank you. We have another question here for you, Shijie. Is there kinetic data available for the SPRM data here?
Yeah. The actually the in the presentation, there's a presentation. I think I mentioned affinity all the time. So, but most of the data given is actually includes the kinetic rate constants like Chi on and Chi off, and also the affinity. So you can get the affinity from the thermodynamic analysis, which is the isotherm analysis, and you can also get the affinity from the kinetic fitting. Essentially, it's the ratio between the Chi off and the Chi on. So yeah, you do get kinetic rate parameters, which is actually the strength of doing SPR measurement.
Great, thank you. Let me see here. Another question that we have here for you, Shijie. Is it possible to obtain binding kinetics and affinity for antibodies conjugated to beads 300 nm size using SPRM 200?
This is actually a very good question. I can take a shot on it, and Marie-Laure, if you have anything to add, you can. To my understanding, there's first of all, there's two things I think here. If you want to measure the binding between the antibody and the bead, or it's actually after the antibody already conjugated to the bead, and you want to do further measurements, of the antibody to other, membrane protein. So in the later case, I think it should work as a normal regular antibody, and our system can handle the size, 200, 300 nanometer size is no problem. But even, however, after conjugated to bees, the size is bigger. I think there must be some effects on the behavior of the antibody, maybe. And so in theory, yes, you can inject those beads with antibodies into the system and measure the binding of the antibody to the other proteins on the cell. But so far, the reality is, we don't have any data on that, but in theory, yes.
Wonderful, thank you. We have another question here for you, Shijie. It says, thank you for the presentation. I'd like to know more about how the suspension cells were immobilized for live analysis with the anti-CD20 MAB. Was CellTac, PLL, or collagen used, and was there any amendments to the regular immobilization protocol?
So the essentially with the suspension cells, I actually got it quite correct. We use different kind of compounds, to coat the chip surface, and use them as like a glue, to grab the suspension cells from the solution and to keep them on the surface. So the commonly used ones, the one you mentioned in the question, including CellTac and PLL and PDL, etc. So for the examples I show in the presentation, the rituximab, actually that was done with the PLL. So we do have protocols to help customers or users to do this kind of experiments.
Wonderful. Thank you so much. All right. And it looks like we have just one more question here. Again, Dr. Wu for Shijie, what is the recommended surface density of cells for SPRM in order to avoid reassociation?
In our experiment, so we culture the cell onto the chip surface. So it's a part of the surface is actually we use like a reference, just like you are doing a conventional SPR experiment. So you have a reference channel, so for some error correction. So usually the recommended confluences of the cell on the chip surface is somewhere between like 30% up to like 70%. So we do have some cell areas and some areas without the cell.
Okay. And Marie, did you have anything to add on that question at all?
No, that has been my experience as well. It's pretty much cell line dependent, but it's anywhere, as she said, between 40 to 70% confidence.
Fantastic. Thank you so much. Thank you again, Marie and Shijie, for your time today and for your important research. We would also like to thank Labroots and our sponsor, Biosensing Instruments, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for all of their interesting questions. Questions we did not have time for today and those submitted during our on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you everyone and goodbye.